Good afternoon, everyone, and welcome to the webinar series, What Lies Beneath? Signature Species of the Delaware Estuary. Today is part 3 of the 3-part series focused on horseshoe crabs. My name is Virginia Vassilotti, and I'm the Schuylkill Action Network Coordinator at the Partnership for the Delaware Estuary. I'll be moderating today's webinar along with Emily Baumbach, who will be managing the Q&A. As well as a couple other PDE staff who will help with any other tech issues that may arise. And so before we get started, I just wanted to go over some tech information for participants. First, all participants are automatically muted with your video turned off when joining the webinar. So you should only be able to see presenters and moderators' videos during this webinar. Second, if you have any questions during or after the 2 presentations today, please use the Q&A feature at the bottom of your screen. There's a toolbar there. You can click Q&A and submit any questions you have. Again, Emily will be moderating that, and we'll have time after each presentation to answer any questions you may have. Third, the chat is disabled for participants. However, moderators will be sharing links and resources with attendees in the chat. And last, the webinar is being recorded in case you would like to view later. And so just some background about the Partnership for the Delaware Estuary, or PDE. We're an environmental nonprofit organization that was established in 1996. We are the host of the Delaware Estuary Program, which is one of 28 national estuary programs designated by Congress to protect America's estuaries. And our mission is to lead science-based and collaborative efforts to improve the tidal Delaware River and Bay, which spans parts of Delaware, New Jersey, and Pennsylvania. And I want to give a special thanks to Ramble for being a sponsor of today's webinar, as well as thanking our 2 presenters for today's webinar. We have Glenn Gavry and Alan Bergenson, who will present in that order. And so what I'll do right now is quickly introduce them, and then we'll jump into some polls just to gauge the knowledge and experience of the attendees here on this webinar, and then go into the actual presentations. So first, Glenn Gavry is the founder and director of the Ecological Research and Development Group, a nonprofit wildlife conservation organization established in 1995, whose mission is the conservation of the world's 4 extant horseshoe crab species, the only such organization in the world. Glenn is a founding member of the International Union for the Conservation of Nature, a horseshoe crab specialist group, and head of the Trade, Industry, and Use Working Group. He is also a founding member of the international series of workshops on the science and conservation of horseshoe crabs held in the US 2007, Hong Kong 2011, Japan 2015, and China 2019. Prior to ERDG, Glenn served as a senior technical coordinator on an internationally recognized oil spill rapid response team, focused on mitigating the impact of oil spills on native wildlife species. Glenn is the mayor of the town of Little Creek, Delaware, where he has served 3 terms. Originally trained as an industrial designer, Glenn has an extensive background in product design and manufacturing. He is also a veteran of the United States Air Force, where he served as an air traffic controller. And then next up is Alan Bergenson, who has over 35 years of experience in industries related by the FDA, including foods, drugs, biologics, medical devices, and cosmetics. He has worked in R&D, QC, QA, and regulatory affairs, and is now marketing as an SME for endotoxin detection. Alan is involved in several scientific organizations, including the chair of the Horseshoe Crab Advisory Panel for the Atlantic States Marine Fisheries Commission, and as the immediate past president of the Capital Area Chapter of the Parenteral Drug Association. He was the chair of the 2004 PDA annual meeting, and he is the co-author author for 3 PDA technical reports. Alan has contributed to USP informational chapter on endotoxin indicators, and Alan has a BA in microbiology from Rutgers University and an MS in biotechnology management from the University of Maryland. And so with that, we'll go ahead and transition into some of our polls that we have here today. So you'll see on your screen, the first one is around what sector you represent: government agency, nonprofit, private entity, university affiliate, K through 12 educator, general interest in topic, or other. So we'll give folks just a couple more seconds to complete that. All right, so it looks like the overwhelming majority is just a general interest of topics. So we're glad to have you here today. Leah, if you want to go ahead and transition into the next poll. And this question is about where you are tuning in from: Mid-Atlantic, East Coast, Northeast Coast, Southeast Coast, Gulf Coast, West Coast, other, or outside the US. Looks like most folks are from our region here in the Mid-Atlantic East Coast. I want to go ahead to the third question. And this is, how did you hear about the webinar? So this is helpful for us if we do additional webinars, how we market the events. There's social media, web blast, email, website, YouTube, from an employer, colleague, from a friend or family, or other. So PD Web Blast is the main avenue for folks to hear about our webinars. That is good to know. And then we have a couple more questions about just your knowledge of horseshoe crabs. So how knowledgeable are you about horseshoe crabs? No knowledge, some, very knowledgeable, or an expert. Great, so it looks like some folks are at least a little familiar with horseshoe crabs, but may not know too much about them. So we're excited to have Glenn and Alan present more in a few minutes. And then, so have you ever seen a live horseshoe crab before? This is simple, yes or no. So the majority of folks have seen a live horseshoe crab. And then our last question: have you ever been administered a vaccine, had blood work done, had an IV of saline, or gotten a shot? And you'll hear more about why we're asking this question in a few. So 100% of people. All right, so thanks, Leah, for sharing that. And with that, I'll go ahead and stop sharing my screen, and Glenn, you can go ahead and share your screen for the first presentation.
Okay. Share, and I think that worked out. My full screen for you?
Yes, that looks perfect.
Okay, good. So I'm on, right?
Yes, go ahead, Glenn.
All right, so for the next 20 minutes, what I'm going to try to provide for you is a global perspective of the world's 4 horseshoe crab species. And then we're going to drill down to the Delaware Bay, where most of you seem to live or visit, and hopefully finish up with enough time to take a few questions before I hand things over to my fellow presenter, Alan. However, before I begin, let me switch this over. Before I begin, I'd like to take a moment to bring your attention to the passing of a very special human being whose contribution to our understanding and appreciation of horseshoe crabs is immeasurable. Dr. Schuster passed away this past May at the age of 100. And actually, you can read a tribute to his life and work in the summer issue of the Estuary News. Okay, so let's get started. We're going to distinguish the difference between a male and female horseshoe crabs. Either big or small, only adults come to shore to spawn. And there are 7 ways to tell the difference between a male and a female. Spawning position being 1, size, curves, claspers, pedipalps, genital pores, spawning scars. So I'll go over a few of those. So We've got the pedipalps on the male horseshoe crab that grab right here on the female horseshoe crab. The Asian species actually have two of these. The American horseshoe crab species only has one. You'll be able to distinguish them between size. You can see the size difference between the male and the female here in the bottom right-hand corner. The curvature is actually this front curvature of their shell. The male horseshoe crab has a more pronounced curvature that allows him to seat more properly on the back of the female. You'll notice that the female, from constant abrasion from spawning, will develop these scars that's in the upper left corner. So spawning scars is a way to identify a female, even if that was the only body part that was visible. They all have 10 legs. Their mouth is in the middle. The chelicera, they have 2 right there. The book gills are right back here. And there are 5 book gills covered by an operculum, which is right there. If you were to lift that operculum up, you would see the genital pores, bottom left-hand corner. And on a female, it's going to be, they're going to look more like a mound where the eggs will come out. On the male, it'll look more like a nipple. Think of it kind of like a valve for the sperm, which will come out. And I think that's all I have on that one. Okay, so this is a picture of all 4 species. So what's the difference between the 4 species? As you can see in the illustration, it's primarily their size, their shape, the front curvature that we looked at before, the length of the spines along the edge of their abdomen, their claspers, the telson shape or profile of it, the shape of their operculum. And this one, Limulus, is in the upper right. That's the only one that lives in the United States. The other 3 species are in South and Southeast Asia. I put their common names right underneath their Latin names. And you can see in this illustration the 2 claspers that it shows on all 3 of the Asian horseshoe crab species, where Limulus only has 1 pair of claspers. And this is the cross-section of their tails. They're all a little bit different. The tail actually in Carcius scorpius is longer than some of the others, the mangrove species. Now, throughout South and Southeast Asia, home to 3 of the world's 4 horseshoe crab species, horseshoe crab conservation is only beginning to take root after centuries of exploiting this marine life primarily for human consumption, and more recently, the production of bacterial endotoxin tests, or it's called TAL because it's made from Tachypleus, versus LAL, which is made from Limulus in the United States. In addition to the harvesting pressure, all 3 Asian species suffer from loss of spawning and nursery habitat. The conservation challenges for the 3 Asian species is complex, for not only is its range extensive, it encompasses a host of countries with very different social, economic, and environmental priorities. And without a centralized governmental body established to regulate and enforce harvesting quotas, it's less likely that meaningful change will occur in time to stop the species' rapid decline. You can see the threats here that we've listed over on the left-hand side. So in South and Southeast Asia, coastal development, coastline hardening against erosion, coastal aquaculture, deforestation for palm oil plantations, and rice paddies have decimated large areas of what was once prime horseshoe crab spawning habitat. This picture in the upper portion is Hong Kong, the lower left is actually Japan, and this is just the ecosystem of the mangroves. The largest threat to the mangrove species, which is the smaller of the 4 horseshoe crab species, is loss of habitat throughout its range. South and Southeast Asia and Asia Pacific contain approximately 46% of the world's mangrove ecosystems. And it's in these regions where the highest rate of mangrove loss and deforestation for alternative land use occurs. And that's the biggest threat to the mangrove species. As for the user groups themselves, the consumption of luxury seafood in contemporary Asia is intertwined with the broader historical and cultural trends. Most importantly, notions of social status through conspicuous consumption, which is linked to the development of the Asian economy and social stratification. Although there are some indications that human consumption of horseshoe crabs in Asia are declining, consumers with higher incomes and educational backgrounds are consuming wildlife at a higher rate. As for the Asian horseshoe crab user groups, throughout South and Southeast Asia, horseshoe crabs are harvested for human consumption and the production of TAL, resulting in 100% mortality. Unlike in the United States, where the American horseshoe crab is collected for LAL, bled, and then released, TAL harvest results in 100% mortality. And the byproducts of these user groups supply secondary markets such as chitin, fertilizer, and traditional Chinese medicine. Picture on the lower left here is a pile of horseshoe crabs being dried out for chitin production. And this picture here in the left is actually a food vendor in Malaysia. The primary stressors for the American horseshoe crab species are loss of coastal habitat, both marine and terrestrial, harvesting for bait in the whelk and eel pot fisheries, and biomedical collection for the production of bacterial endotoxin test called LAL. The southernmost range of the American horseshoe crab falls outside of the jurisdiction of the Atlantic States and the Atlantic States Marine Fisheries Commission. And although it's protected by the Mexican government, it continues to be threatened by loss of habitat and poaching for bait in the octopus fisheries. Unlike the 3 Asian horseshoe crab species, the American horseshoe crab has come a long way from the days of unregulated harvest and population decline. After 23 years and a series of coast-wide science-based harvesting restrictions, the American horseshoe crab shows signs of recovery throughout most range. It's important, though, to remember that horseshoe crabs are slow to mature. Consequently, it can take years to see the results of a regulatory policy. Another important point to remember is that horseshoe crabs are genetically diverse throughout their spawning range and vulnerable to local extinction from unsustainable harvesting. Consequently, state and regional harvesting decisions in the United States are evaluated against the health of the population where the harvest occurs. Like in Asia, loss of habitat and its cause is our species' greatest threat. The American horseshoe crab does not spawn along our ocean coastline. The surf is too high energy, which easily strands them upside down during spawning and erodes their nest and exposes their eggs to the elements and predation. Offshore is too deep, cold, and not a very suitable nursery habitat for the juveniles. So it is the bays, the backsides of barrier islands, the tributaries where horseshoe crabs spawn. The surf is low energy, nearshore areas are warm, shallow, have an abundant food resource for the juveniles before they grow and venture into deeper waters. The reason why the Delaware Bay is one of the best spawning areas in the world is because it is shallow with an optimum temperature and has over 150 miles of low energy beaches. This picture of population density at Ocean City is quite remarkable, actually. It's a great aerial shot for you to see. In the interest of time, I'm going to just skip the discussion on this. This is one of ERDG's stranding mitigation projects, but however, if you're interested, there's information posted on our website and our Facebook page. This is the north end of Pickering Beach, and this is what it looked like from habitat neglect, and this is what it looks like after we created our barrier. In the US, from the late 1800s to the early 1900s, the American horseshoe crab was harvested unsustainably for fertilizer and livestock feed, depleting the resource by the 1940s. After a brief recovery as the fertilizer and livestock feed industries moved away from horseshoe crabs to alternative products, 2 new user groups emerged: the biomedical industry in the mid-1970s and the whelk and eel pot fisheries in the mid-1990s. In 1997, the Atlantic States Marine Fisheries Commission was tasked to develop a coast-wide horseshoe crab fisheries management plan that balanced the various stakeholders' interests and to ensure sustainable population throughout its spawning range. ERDG has always treated the horseshoe crab species as a non-game species in an environment where it's mostly viewed as a commodity. On highly productive spawning beaches, such as those within the Delaware Bay, buried egg clusters, past and present, get disturbed during spawning activity and churn to the surface, creating bands of eggs along the high tide line. No longer viable, they become food for a host of nearshore animals and migratory shorebirds, most notably the red knot, who depends on these eggs to refuel en route to their Arctic breeding grounds. Many of these birds have traveled as far away as the southern tip of Chile. Without this natural resource, their ability to reach their Arctic breeding grounds with sufficient energy reserves, it would be severely compromised. This picture on the lower left actually is a beach up in the northern part of Pickering Beach, which sometimes has an incredible amount of horseshoe crab eggs that have been turned upside down. The Delaware Bay is home to the largest spawning population of horseshoe crabs in the world, which is currently estimated to hold 25 to 30 million adults. The success of this location is due in part to 150 miles of low energy sandy beaches, shallow shore nursery areas with an abundant food resource, optimum salinity, and temperature. In 1998, to help preserve this remarkable habitat, ERDG began a backyard stewardship community horseshoe crab sanctuary program designed to engage coastal communities behind the conservation of this remarkable animal. In 2001, the National Marine Fisheries Service established the Carl N. Schuster Jr. Horseshoe Crab Reserve, prohibiting the harvest of horseshoe crabs in federal waters within a 30 nautical mile radius off the mouth of the Delaware Bay, which is habitat to a large percentage of the Delaware Bay's subadults and adult crab population. In addition, there are several ongoing projects around the bay geared towards preserving and/or restoring essential horseshoe crab spawning habitat. This large area on the Delaware side of the bay is Broadkill Beach, which is actually directly opposite across the bay from Cook's Beach in New Jersey. The reason why this is such a massive undertaking is this is the result of the dredging spoils from the Delaware Bay deepening project by the Corps of Army Engineers. So there's contract to keep this going for many years into the future, actually. Since 1999, the American Horseshoe Crab Tagging Program, managed by the U.S. Fish and Wildlife Service, has recorded 362,500 horseshoe crab tags from Maine to Florida, with over 44,700 reported sightings. This is an important program that helps us understand the movement of these animals and their longevity. So if you see a horseshoe crab with a tag, report the tag number. Do not remove it unless the animal is dead. You can call it into a website, and when you do so, you'll get a tag, you'll get a certificate that actually tells you when the animal was tagged and any time that it was resighted. So some of the times you can track these animals from one beach to another. We had one this summer that was tagged by Lanza in Ocean City, Maryland, then made its way into Rehoboth Bay, stayed there for a few years, and then ultimately made its way up to Pickering Beach, northern part of their spawning range in the Delaware Bay on the Delaware side. So if you see these, definitely call them in. Horseshoe crab spawning season for the American horseshoe crab varies according to latitude, but generally peaks in May and June, with peak spawning occurring on the evening high tides during the full and new moons. The adults seek beaches that are at least partially protected from the surf within the bays and the coves. By the beginning of the spawning season, adult females, which are 11 to 12 years old, will have developed around 80 to 90,000 eggs. She will come to shore on successive high tides, laying 4 to 5 clutches of eggs, each about 4 to 8 inches deep with each tide. Each of these golf ball sized clutches contain 4 to 5,000 eggs. Adult males who are 9 to 10 years old, who are about 20% smaller than the female, use a specially developed appendage to clasp themselves onto the back of the females. Other males will group around the females, adding their sperm to the water that flows over the eggs while they're being laid. If you look at this picture here, if you were to see 2 horseshoe crabs, male and female, it would be the upper left. If you were to see 2 males to a female, it would be this optimum position, and then this one would be over this vent there, which allows their sperm to wash in over the eggs. If you were to see 3, you guarantee it's going to look like this over both vents. And then beyond that, they just satellite around the female, all releasing their sperm, all of that sperm being washed in and out, in and over those eggs, so the water passing through those vents. Fortunately, horseshoe crabs have a very high hatch out rate, which is important because just about everything eats them. The juveniles will live near the nearshore areas for several years, going deeper as they get older and larger. Very few will reach their first birthday, and fewer still will reach adulthood. By the 4th day after fertilization, rudimentary appendages can be seen in the developing embryo after the outer chorion layer ruptures. The horseshoe crab juveniles hatch out by the 14th day and begin their cycle of growth and molting that will continue until they reach their adult size 9 to 12 years later, after which they will no longer molt for the rest of their life, estimating to be another 8 to 10 years. This picture on the lower right-hand side is a horseshoe crab at 1 year old. So you can see at the size that these animals are, just about everything is going to eat them. Again, for the interest of time, I'm going to skip the discussion on the internal anatomy. However, if you're interested, you can visit the anatomy section on our website. One thing, though, that I would like to point out, because it'll probably be in Alan's slide, is the heart of the horseshoe crab is actually quite large. It's this long area here. And when they do bleeding, they do a heart stick through the center area, that hinge area between the prosoma and the opisthothoma is where they do that. They go right into the sinus cavity of the heart, which is right about there. It's a large heart. 10% of the spawning horseshoe crab population will get stranded upside down or right side up. And in fact, more animals die each year in the Delaware Bay from being stranded or harbored within the bay for bait and collecting for biomedical purposes. Waiting for the next tide is also always not a good strategy. The tide doesn't come and the sand dries out, they die. The path to death begins where there is no longer oxygenated salt water passing over their book gills and their body temperature begins to elevate. At that point, survival is a matter of conditioning and time. Often animals are returned to the sea moving, but they do not survive. You can see in this picture, this whole area here was a higher than normal high tide, probably a full or new moon high tide the evening before. And because of the overall density of spawning on this beach, a lot of these animals that are up high didn't get back to the water as it started to recede and decided to hunker down and wait for the next tide, which in this case will never come. So this water, this beach area will dry out, their book gills will dry out, and they'll perish in place. To address horseshoe crab stranding mortality and to encourage the general public to get involved with the conservation of this species, ERDG started our Just Flip 'em program in 1998, which is now in use throughout the world where horseshoe crabs come ashore to spawn. No experience or training is needed, just an open heart and a gentle touch. So if you're walking along the beach where horseshoe crabs spawn and see an unfortunate critter stranded upside down, for one reason or another, offer a gentle hand on the side of its shell and just flip them. It will find its way back into the water. Horseshoe crabs are very gentle, they won't hurt you, so there's no reason to pick them up by their tail, which would also injure them. And more recently, a similar program called Return the Favor was started in New Jersey to address the problem of New Jersey beaches, many of which are closed during the migration of the shorebirds. So they're restricted public access. Through this program, volunteers get trained and they're allowed access to these restricted beaches at certain times of the day to help stranded horseshoe crabs. So you can make a difference no matter where you live, if you live on the beach or if you're visiting the beaches. Between these two programs, there's something for all of you to do, and I can assure you there's no shortage of horseshoe crabs that are stranded. Please keep in mind that there's more to horseshoe crab conservation than what goes on in the Delaware Bay and the migratory shorebirds that visit each year. There are a host of issues that threaten these remarkable mariners from Maine to the Yucatan, from Japan to India. And the work is only possible through the dedication of countless scientists, nonprofit organizations, communities, and individuals throughout the world who are working together to overcome the ignorance and indifference that have threatened the survival of these remarkable living beings. Last year in China, at the 4th International Workshop on the Science and Conservation of Horseshoe Crabs, the International Horseshoe Crab Day was established, which will be held every year on June 20th. This year, because of the COVID-19 pandemic, most of the celebration was online, but hopefully next year you'll begin to hear voices from around the world speaking out in celebration of this remarkable mariner. And with that, I will pause and take any of your questions. Do I have some time remaining?
Okay, great. Thanks, Glenn. That was a very
Yes.
informative presentation on the American horseshoe crab, as well as the Asian horseshoe crab species. Lots of really great info in that talk. 25 to 30 million horseshoe crabs in the Delaware Bay. Wow. So we do have a few minutes to ask Glenn some questions. If folks want to type any questions in the Q&A box, I think we had a few come in already. So let me take a look.
I tried to click on that, but it doesn't open on my screen. I'm not sure.
No problem, we have them.
Okay.
So here's one. Why don't horseshoe crabs spawn in the Chesapeake Bay?
Actually, they do spawn in the Chesapeake Bay. There's a number of areas in the bay. Actually, if you go on our website, there's a section called Horseshoe Crab Sightings. And if you click on that, it will give you every state along the Atlantic coast. It'll give you the Yucatan Peninsula, and it'll give you every country in Asia where horseshoe crabs spawn. And if you click on the country or state that you're interested in, it will show you the locations where people have recorded, where we have recorded, or people have called in sightings from those areas. And there's a number in the Chesapeake Bay, going actually all the way up to the Chesapeake Bay Bridge.
Wow, okay, great. And that's on the ERDG website?
Yes, under horseshoe crab sightings. They can click there and find a location that's near them, and then they can go visit that and look at horseshoe crabs. Well, they can't do it now, but but next year they will be able to.
All right, very cool. Another question was, what number do we call to report a tag sighting?
Don't know it off the top of my head, but I don't need to know it because it's on the tag. The tag will have the number to call in. It'll have a website if you want to use that. The website is actually the better of the two. Sometimes the lines are busy. Whereas the website, you can put in your information there. They get back to you pretty quickly within a day or two, and they'll give you the information about the animal you saw. It's a really neat program. It's not only scientifically valid and helpful, but it feels good for the general public to get the feedback of what they saw on the beach.
All right, great. I'll pick a couple more. We have a lot of really great questions coming in. Here's one. So when you flip horseshoe crabs over, should you move them towards the water or move them or just toss them into the water?
Challenging question and mixed answers. I think by and large, for the most amount of people, just flip them over and leave them in place. Usually, if you're going down the beach and they're stranded from the night before and they're well within the normal high tide line, as you go down the beach and you turn around and look behind you, you'll see them all kind of marching into the water on their own, which is kind of a cool thing to see. So you really just need to flip them over and leave them in place. However, if it's mid-afternoon, the sun's baking and they're higher up on the beach from a higher than normal high tide, and you know that the next tide is never going to get to them, if you're so inclined, you could pick them up and bring them down to the water's edge, because they will die there.
Okay, great. We do have a couple questions coming in about horseshoe crab blood, but I might leave those off for coming back to after Alan's presentation.
Yep.
One question came in. It said, can you expand on how chitin is used from the horseshoe crab?
Chitin is actually in just about every product well, not in every product you can think of, but it's in quite a lot. It's a lot in cosmetics, it's in toothpaste, it's in filtration systems. It's used as a filler in quite a lot of materials. A good bit of the chitin is made from shellfish like shrimp, and most of the chitin manufacturing is done outside of the United States and throughout Asia, primarily because of the chemical process needed to separate the chitin from the shell. Creates a material that's difficult to environmentally dispose of. So consequently, chitin production tends to be done in areas where environmental regulations tend to be more relaxed.
Great. All right, I'll just do one more question, then we might be able to come back to them later on in the webinar, or we'll put Glenn's email address in the chat as well if people want to reach out to him directly. So are there public events where we can view horseshoe crabs spawning and raise awareness of these wonderful creatures?
The best public events is to go to the beach. Dr. Schuster always said, if you want to know about this animal, you go to the beach where they spawn, you get down on your knees and you lay down on your belly and you look at them eye to eye. So that's one of the reasons why we have the sightings area on our website. So you can find a beach near you, or if you're visiting an area, you can find a beach where they may be in that area that you're visiting, and go there during the right time for spawning and watch these animals spawn. Help the ones that are stranded upside down. Report tags that you see, clean up the beach while you're at it, and most importantly, educate others as to why you're doing what you're doing, so that you tend to pass that along to somebody new.
Great. All right, we'll ask just one more, as I think we have enough time still. When in the lives of the horseshoe crab, is their gender determined, and how? Is it genetic, environmental?
Unlike turtles, where that's usually developed by temperature and in their eggs, we don't think that that's the case with horseshoe crabs. But you can determine a horseshoe crab's sex really early in its life. All you have to do is be able to lift up the operculum, and you can see the difference between their genital pores. So even if you've got an animal that's relatively small and easy to handle, it could only be maybe 3 inches across. It could even be a molt. If it's a molt and everything's intact, including the operculum, all you have to do is soak it in warm water to soften it up, lift up the operculum, and look at those genital pores. You won't be able to tell from the other signs that we talked about earlier, because they're not large enough to have developed those cues. But the genital pores will be a surefire way of doing it.
All right, great. Thank you so much, Glenn. And with that,
Pleasure.
I'm going to turn it over to Alan's presentation.
I'll share a screen. How do I do that? Stop share. Okay.
Okay, are we all set for mine?
Oh, good. Take it away.
Okay. First, I'm Alan Bergenson, and I work for an LAL company, and I'm also the chair of the ASMFC Horseshoe Crab Advisory Panel. And I usually start my presentations with this saying, because it applies. In the beginning, Greeks considered fever to be beneficial rather than hazardous, and Parmenides, an ancient Greek physician, said, "If I only had the means of creating a fever artificially, I would be able to cure all illness." Well, that's not always true. So today we're going to talk about what is endotoxin and why do we care? How do we test for it? What products are tested? Why horseshoe crabs? Horseshoe crab population information, risks to horseshoe crabs, and the current population status from the Atlantic States Marine Fisheries Commission. So first we have to go through a few descriptive terms, some definitions. Pyrogens. What are pyrogens? They are any substance that can cause a fever. Endotoxin is a component of the outer membrane of certain bacteria, and they are lethal in nanogram, which is a billionth of a gram amounts if injected. Now think of it. That's 1 billionth of a gram. There are 28 grams per ounce, so that gives you an idea of how lethal this is. And it's one of the most lethal, or one of the most potent toxins that's known. And then there's lipopolysaccharide, which is just a chemical name for the purified endotoxin. So looking at that gram-negative bacterium, you can see on the bottom is the lipid bilayer. Then there is a peptidoglycan layer, which is a kind of a hard layer. And then on the outside, there is another lipid bilayer, but there's also other things. If you look to the right, you will see the orange ovals with the blue squiggly lines. That is a lipopolysaccharide molecule. And then part of those are the polysaccharide tail, which is used to identify bacteria in many cases, and lipid A, which is the toxic part of the lipopolysaccharide. And if you're interested in the chemistry, this is a representation of what a lipopolysaccharide molecule is. Again, that lipid A moiety is all the way on the right. It's a diglucosamine with diphosphorylated with a long chain hydroxyl fatty acid tail. So endotoxin contamination. Right now, research demonstrates that important principles in connection with endotoxin is that it's ubiquitous. It is everywhere. It is on the surface. It surfaces your environmental surfaces, your computer desktop, the water you drink. I've measured my own endotoxin concentration of my well water, and it's between 5 and 50 endotoxin units per milliliter. But if you drink it, it won't hurt you. It's heat stable. It's associated with injectable products. Even if you steam sterilize a product, it does not remove the endotoxin. So you can have a perfectly sterile product, but it's still pyrogenic because of the endotoxin. So in order to manufacture pharmaceutical products, you have to use sterile production procedures and adhere to them. And like I said, it's only associated with gram-negative bacterial contamination. So the effects of endotoxin on humans and animals, the least worrisome is fever. But then with a slightly higher concentration, that same person, if the endotoxin finds its way into their bloodstream, will go into shock. A little more, they'll go into a vascular collapse. And a little more, and they'll die. And this is in nanogram amounts if it is injected into your bloodstream. So the one thing that we do now, and this is one of the questions that we posed earlier, how many people have ever had an injection? Everybody has. But and we don't think anything of it. We're pretty cavalier. Yeah, I had to get a shot. But in the early days of injection, it was risky, because you could develop something called injection fever, and it was highly probable that you would get an injection fever, and a certain amount of those people would get very sick and die. And there was no way to know whether the substance being injected into your veins was pyrogenic. So in the early days, doctors had to weigh the risk of injection fever with the prognosis of the patient. You know, it's benefit versus risk analysis. Because, like I said, there was no way to detect pyrogens in injectable medicines until 1941 and USP 12. So let's go through the story of Limulus amebocyte lysate. And our story starts with the little bunny you see on the left-hand side. The original pyrogen test was the rabbit test. Why? Because it produces a pyrogenic response when you inject the rabbit. The other species are not so predictable. They looked at many other animal models, and there was the two that they came down to was rabbit versus dog. Rabbits gave false positives. And one thing about rabbits is if you are doing a test on a $1 million batch of your favorite drug meant for injection, and somebody walks into the room and scares the rabbit is notorious for spiking fevers, and they you fail your batch. Dogs, on the other hand, give false negatives. And that's bad. You want to be able to detect a pyrogenic response if it's there. If it's a false positive, the patients are still safe. And one thing about the rabbit too is that it's got a similar pyrogenic threshold that humans do, and that's approximately 5 endotoxin units per kilogram. So, discussing the rabbit pyrogen test, in 1941, the USP Expert Committee on Revision authorized a collaborative study involving the US Food and Drug Administration, the National Institutes of Health, 14 large pharmaceutical companies. And the results of the study led to the rabbit pyrogen test being published in 1942 in USP 12. And the test is still available, and it has not really changed since that time. And it's listed as Chapter 151 Pyrogen Test in the United States Pharmacopoeia. So let's look at the rabbit test. You have to weigh the rabbit before you test. You transfer to a restraining box. You insert a rectal probe, a temperature sensor. You warm all the solutions to 37 degrees. You inject 3 rabbits in their ear veins, and you look for a rise in temperature. And here are the rabbits undergoing a pyrogen test. They look like they're having a real banging time there, don't they? And so it was the horseshoe crab that got away from this type of testing. And so we now have our friend, the American horseshoe crab, Limulus polyphemus. So where did the idea that horseshoe crab blood extract could detect deadly endotoxin in injectable products? Well, it was known since W.H. Howell of Johns Hopkins back in 1885 that horseshoe crab blood clots. And during the 1950s, Fred Bang from MBL elucidated the pathway of exactly how the blood clots. And later, Dr. Bang and Jack Levin developed a method for detecting endotoxins using an extract of the white blood cells called amebocytes, and the product was called Limulus amebocyte lysate. In the 1970s, the LAL test was recognized by the Food and Drug Administration as an alternative method to the rabbit test. You still had to do the rabbit test, but they were allowing you to begin using LAL. And due to efforts of people like Don Hochstein, Ed Seligman, and Jim Cooper at FDA, the LAL test became the method for detecting endotoxin contamination in injectable products and medical devices. So LAL, like I said, is derived from the blood of the American horseshoe crab. It's currently the only test available, asterisk, to detect endotoxin in human parenteral and animal injectable pharmaceutical preparations. As I said before, endotoxin is everywhere, and you have to try to keep it out of your bloodstream. Endotoxin testing, or pyrogen testing, of all parenteral medicines is required by US law, and it's actually required by law around the world. The test method is specified by all the major pharmacopoeias, and the pharmacopoeias are the official test methods and specifications for pharmaceutical production. So that means the United States Pharmacopoeia, the European Pharmacopoeia, the Japanese Pharmacopoeia, the Chinese Pharmacopoeia, they all specify LAL testing for pharmaceuticals. And hundreds of millions of parenteral dosages are administered annually. And here's something that I really want everyone to have as a takeaway. Every person on Earth who has ever had an injection owes the safety of that injection to the horseshoe crab. They no longer have to worry about injection fever, getting a fever, undergoing vascular collapse, going into shock, and dying. And right now there are 7.8 billion people on Earth, and at some time, every one of those people will have an injection. So what is tested? Like I said, all parenteral, which means injectable products, including vaccines, flu, childhood vaccines, etc. And even you heard a lot about the development of the COVID-19 vaccine. That will also use LAL to test. Intravenous drugs, so if you've ever had antibiotics, if you take insulin, if you take interferon, if you any type of injection, it has to be tested. And if you've ever had an IV saline, so you've gone to the hospital and the doctor thinks you look a little dehydrated, and they hang a liter bag of saline on the holder, that fluid was tested for endotoxin using LAL. But not only injectable drugs, but also implantable medical devices, including replacement joints, sutures that are meant to stay inside the body internally, stents. So if you've ever had a cardiac stent or a biliary stent, they are all tested, and also radiology imaging fluids. So if you've ever had an ECG or anything like that, you're going to have that product tested using LAL. So what kind of LAL products are available? There's the gel clot test, and that goes off the basic work of Fred Bang and Jack Levin, there's a endpoint chromogenic test. So instead of going through that reaction and getting a gel clot, you will actually get color produced. There are also now automated assays where you look at the development of turbidity rather than a gel clot. So you're looking at turbidity development over time, and once it goes through a threshold, you can measure that. And the same thing for chromogenic assays. You look for development of a color rather than turbidity or clot over time. And the reaction sequence is simple, it's elegant, and it's absolutely amazing. First, you start out with a proenzyme, and endotoxin interacts with that proenzyme from the amebocytes to give you an active enzyme. And then that active enzyme works on the clotting protein to give you a clot. So in this picture, you can see, if you look at that little tube there, you can see at the end, that is a gel clot. So if you gently invert the tube and the clot stays at the end of the tube, you have a positive. That means there is endotoxin in that sample. If you turn over the tube and you get a wet hand, that means it's negative. And you don't have endotoxin present. And this graph shows how all of those different assays that I described are all start with the same thing. They start with the gel clot test with factor C all the way down to the recombinant product with recombinant factor C. They all, factor C changes factor B and works on the pro-clotting enzyme, giving you a clotting enzyme, and you go from coagulagen to coagulin, giving you a clot. If you look for the kinetic chromogenic assay, once again, factor C works on factor B, works on the clotting enzyme, pro-clotting enzyme, giving you a clotting enzyme, and which cleaves the chromogenic substrate and giving you a color. With the recombinant product, you've got recombinant factor C that doesn't use any of the signal amplification steps that the natural product has, and so the activated factor C, because of the endotoxin, will cleave a fluorogenic substrate, and when you excite that substrate at 380 nanometers, you will get light produced or fluorescence. So going back with the rabbit test, when quote unquote new LAL test first came around, it was quickly found to be at least comparable to the rabbit test, meaning you're more likely to get an equivalent result or a positive than you are a negative. So you will detect the endotoxin. And one thing of interest to note, in 40 years of the endotoxin assay using LAL, there has not been one product released to the market that was later found to be pyrogenic due to endotoxin contamination. That is a fantastic track record, especially when you look at the track record from before, where your physician was literally taking your life into his hands by giving you an injection. So due to improvements in the manufacture of LAL, the test is now 10 to 50 times more sensitive than the rabbit test. And it's important to note that the rabbit test measures a dose response, where the LAL test measures the concentration of endotoxin in a product. So the advantages that the LAL test has over the rabbit test: it's fast, it's 60 minutes versus 3 hours. It's a lot more sensitive, like I said, up to 50 times more sensitive than the rabbit test. It's a lot less variable, and that's what I had mentioned before about somebody walking into the room while you're performing a rabbit test, scaring the rabbit, and the rabbit spikes a fever. You have much less false positives, and once again, scaring the rabbit. It's much less expensive. Right now, if you tried to go back to the rabbit test from the amount of testing that is done in the pharmaceutical industry, one thing I like to say, there ain't enough rabbits in the world to go back to that and do that type of testing. And it's also an alternative to using live animals as a testing model. Nobody wants to do that anymore. But one thing about the LAL test, testing can be now done on raw materials, water, in-process samples, and final product. You don't have to wait until the final product is made to test to find out, is this pyrogenic or not? Because you've built quality into your product. So if you have no pyrogenic raw materials, no pyrogens in your water, no pyrogens, no endotoxin in your in-process samples, as you go through your manufacturing process, you're just about guaranteed that the final product will be non-pyrogenic due to endotoxin. And that's building quality into a product rather than final products. And that's why I said before, there's a 40-year track record of testing with LAL, and a positive product has never been put on the market due to a false negative test. So this is a slide that is similar to what Glenn had performed or showed before. You can see on the right hand of the screen, the Asian species in orange and purple. And then on the right hand side of the screen, you can see the blue areas, which is the North American horseshoe crab, Limulus polyphemus. So, as Glenn already said, the Asian populations have been severely depleted due to lack of regulation, pollution, lack of spawning habitat, use for many different reasons, including fertilizer. It's just not a sustainable situation whatsoever there. And the North American horseshoe crab distribution, Limulus polyphemus is distributed from Maine to Florida. And as Glenn showed you before, the largest concentration of Limulus is found in the Delaware Bay region. And one thing, the horseshoe crab population had declined seriously in the early 1990s due to essentially a lack of regulation, overfishing, and because it's so important to the migrating shorebirds, such as the red knots, it really needed to be protected. So with the regulatory limits, horseshoe crab harvest for bait is regulated by the Atlantic States Marine Fisheries Commission. And when you get the slides, that is a live URL, so you can just go there yourself if you wanted to and see how the horseshoe crab is regulated by the Atlantic States Marine Fisheries Commission. So in 1998, the Horseshoe Crab Commission Fisheries Management Plan was developed. It imposed state quotas on horseshoe crab harvest for bait, although horseshoe crab collection for LAL came under a biomedical exemption because horseshoe crab use was regulated by the United States Food and Drug Administration. However, the ASMFC set up a limit of 57,500 maximum industry mortality based on a 15% theoretical calculated mortality. The actual mortality that we report, that all of the manufacturers of LAL report to our various state representatives, is much, much lower than that. So we report that regularly to the Department of Natural Resources in the states that have horseshoe crab blood collection facilities. And those facilities are monitored by the state's DNR and reported to the Atlantic States Marine Fisheries Commission. And one thing I should say is that the Department of Natural Resources also do their own monitoring of the operations, and they compare their numbers to the numbers that are reported to the various DNRs. And if there is a discrepancy, the LAL or the LAL manufacturing company risks losing their license for collecting horseshoe crabs to produce LAL. That's not something anybody wants to have happen.
Alan, sorry to interrupt. You have just about a minute or so left to wrap up, if that's okay.
Oh, I do? Okay. Well, this is the The bleeding, the landings you can see on the left-hand side of the screen. There was a lot of landings that went down, and then the orange lines are biomedical collection. But one thing with the bait landings, it's 100% mortality. With biomedical collection, it is not. The mortality, the estimated mortality, is those green lines, and it shows a very low mortality. So to wrap up, the major sources of mortality in 2019 was bait harvest with 100% mortality, according to the ASMFC benchmark stock assessment and peer review report. The bycatch from other fisheries was about 50% mortality, but it's a much higher fishery because it's every other species that you fish for on the bottom. Erosion prevention, as Glenn showed, with riprap and seawalls and the like, the animals get trapped in there or they're not allowed to spawn on the beach. There's stranding with an estimated 10% mortality of the entire population annually. And as Glenn said before, the best thing anybody could do when walking on the beach is just turn the crabs over. And I've been doing this since as Glenn knows, I've been doing this since I was 3 years old fishing with my dad. You just turn them over and I put them on the edge of the water and in they go. And one thing the ASMFC peer review report said that the biomedical mortality is really not really affecting, it's inconsequential to the horseshoe crab population, and they should receive less focus in the future. So the current population status is done by the ARM. I won't go into how that works, but it sets quotas every year. And this was the 2009 quotas. And even though the ASMFC in Massachusetts set a 330,000 horseshoe crab quota for bait, Massachusetts chose to do less, and most other states did too. The current status, back in 2009 when they did the assessment, the Northeast was not doing very well, whereas New York was okay. Delaware Bay was stable. The Southeast is great, and because they don't have any bait collection in the Southeast. And coastwide, everyone was doing good. However, in 2019, the New York Bight area is doing very poorly. And it's one thing to note, there are no LAL production facilities in the New York Bight, and nobody collects horseshoe crab blood in that area. So it's not due to that. And one thing to wrap up, and I want to pay tribute once again, as Glenn did so very well before, is the recognition of a giant of a man, the Carl Schuster Horseshoe Crab Wildlife Refuge. It's just a fantastic tribute to a fantastic man. And with that, I say thank you. And if you have any questions, I'll be happy to answer them.
Thanks, Alan. And just in the interest of time, we're not going to be able to get to Q&A today, but Emily's going to include your email in the chat just so folks can follow up with you if they have any questions. And just real quickly to wrap up, I'm going to share my screen for just some final slides to thank our participants. So first, the reason why we really care about this work is because it relates to the comprehensive conservation management plan for the Delaware Estuary. And so we have, I think my slide went away, there we go. We have a strategy in there that is specifically related to protecting and restoring horseshoe crabs in their habitat. We're interested in hearing any success stories that you may have related to horseshoe crabs. So you'll see the link below that you can share those success stories. And with that, I just wanted to thank both Glenn and Alan for presenting today on horseshoe crabs. They were two great presentations that will be live after the webinar, and want to thank also all the attendees for tuning in today. And if you're able to, please consider making a donation to PDE to help us achieve our mission. So thanks again, and we'll have the recording available either later today or tomorrow.